We have a really great special guest today, Catherine Cunningham, who is joining us from UT in Galveston, and is going to give us a great presentation on defining the path to addiction therapeutics. This is exactly what I asked John about, a little bit of context for this presentation. As we build the new institute, one of the areas of emphasis is going to be in the area of addictions, and specifically how do we translate some of the really great work that some of you are doing in more of the basic sciences into treatments. And one of the advantages we have is I think we have some pretty strong basic science work. We have, on the other end of the translational spectrum, we are the only psychiatry department for a 5-state region. We have very well established, very good relationships with not just clinics, here in Seattle, but we have, for example, a new grant, 16 on how much? 16, 18 million, something like that. $14.8 million from NIH to work with clinics all over the country to see can we help support medication-assisted treatment for opioid use disorders using a collaborative care model. So those are the kinds of things that if we had treatments, if we had new treatments, we would be actually in a pretty great position to rapidly bring them to scale. So I think that's a tremendous opportunity that we have here, and we have a big gap. And in my mind, that gap is, how do we move from what you're doing in the lab to what we now can do, with a whole bunch of clinics. I think we need more treatments, we need better treatments. And I said to John Lohmeyer, who I would turn to with these kinds of things, that John, I need a couple of people who can give us some really great advice on how do we fill this gap? What do we do with this gap in the middle? How do we translate to human treatments? And the first person he mentioned to me was Catherine. So I am super excited that she took time to come and visit with us. She met with some of you today. I look forward to her presentation, and I'm going to ask John to finish the introduction. But thank you so much for coming.
Well, thanks, Jürgen. I made a few notes because my first thought was, well, it's obvious we're having Catherine come to visit. She's the right person to talk on this topic. But let me tell you more about her. She's the Chauncey Leake Distinguished Professor of Pharmacology, and she directs the NIDA Center, Center of Addiction Research at the University of Texas Medical Branch in Galveston. And she graduated from the University of St. Thomas in Houston and then completed her PhD at the University of South Carolina. Then after a postdoctoral time in Galveston, she joined the faculty and moved up through the ranks and has had a vast number of leadership positions at UTMB in Galveston, but also many other organizations. She's published over 160 papers, and she's received all kinds of awards from major organizations that have to do with pharmacology and addiction, like ASPET and the CPDD and the International Society for Serotonin Research, which is where we met when we were in England at one time. And she gets these awards for a couple of reasons. One is the important role that her research plays in molecular pharmacology of addiction. The second level that's important is that her work really tries to stand, and her collaborations stand from medicinal chemistry and rational drug design through animal research to human studies, and she has collaborations that just span this whole range. And that might be enough, but she's also incredibly important in the leadership of professional organizations and in mentorship and in encouraging women and men to progress in their careers. And she's just a fantastic leader in our field. And I thought that she had lectured on every continent, but then I found out we have still Asia and I think Antarctica to cover. And I suggest that you wait on both right now. And so we're fortunate that she's here today to teach us today. And let's all give a thank you to Catherine for joining us today.
Thank you, Jürgen, for your comments, and thank you, John, for such a nice introduction. Is there a light control?
Yes.
That's a little dark. Can you hear me? Okay, it was great I really enjoyed my time, but well, yesterday too. I really enjoyed the whole event. I mean, it might have been a little dull yesterday after flying all that time, but I really have connected with a number of people here. We've had talks about some pretty amazing opportunities that this campus and the city has to offer. And this is a kind of different talk than I have a couple of different versions. This is not my molecular neurobiology talk. This is my translational talk. So I hope it actually captures sort of the place we've been and how we've gotten to the point of trying to define this path. And as everybody in here knows, either from the basic translational or clinical sciences in psychiatry and related disorders, we have a big problem and we need to deal with it. And that problem actually really reared an ugly head in the 80s when cocaine was really all the rage. And this is just a collection of some of the 1980s and the early into 2012 covers of Time magazine. I don't even know if Time magazine still exists anymore. But these were very compelling problems. This is a very compelling problem, which we address with this. Just saying no. And it was all in the best intentions. I'm not saying anything negative about it. It was the best intentions. But somehow, this did not capture what we needed to do. Hence, we're standing here right now with a major drug overdose problem. The leading cause of death in 2017 for those under 50 years of age were drug overdoses. How did we get here? It's an unbelievable problem to me. Of course, we know that opiates are the primary driver right now. And these numbers here indicate that 2016 is in blue and the predicted 2017 is in yellow. So you can see it's not dropped. It seems to be dropping in 2018 in certain parts of the country. But that's not the only problem. It's not just opiates. In the 80s, we dropped the problem with cocaine. Look at this rise here. Still rising. And methamphetamine, still rising. In the state of Texas, we don't have an opiate problem. We have a meth and cocaine problem. And now we're getting a fentanyl problem coming up from Mexico. So we have not fixed the problem. And is it really about the drug that is being abused, or is it a larger issue of vulnerability, and what are those vulnerabilities? So ultimately, those people who survive overdoses, this signals the growth of a problem in opiate use disorder. And I was reading a market survey the other day, which is the financial people trying to decide how much this is costing the healthcare organizations in the country and how much is it going to cost us in 15 years. Right now, there are about, for opiate use disorder, in 2017, there were about 2.5 million diagnosed cases of OUD, costing about 400,000, I can't even think of these numbers. 400 million, I think, a year for treatment. Those people that get treatment. 15 years from now, there'll be 31 million people diagnosed with OUD if we don't do anything about this. And that will cost us in the billions and billions of dollars. So how do we deal with this issue? And where are we going? Everybody that's in the world of addiction science here will recognize this picture. Originally evolved from George Koob's very prolific brain in terms of neurocircuitry. This picture is actually a variant on it from Volkel, Koob, and McClellan in 2016. The cycle of substance use disorder, the binge intoxication, withdrawal, negative affect, preoccupation, and the continuation of this cycle. We have uncovered much about the neurocircuitry involved, and we are looking at many of the behavioral targets that we are engaged in this cycle in animals and in humans. So the reward circuit, drugs hijack that reward system, habit kicks in. That involves the basal ganglion and then the resulting intensive desire for drug. Targets that might be important in terms of reversing this is obviously the rewarding response to drug. But now there's work here and elsewhere to suggest that we must look at resting state patterns in fMRI, try to understand vulnerabilities to the the use of drugs to begin with and the rewarding properties of drugs. The stress circuit, of course, we have some of the leaders in the room, like Charlie, in the stress circuit, which is a maladaptive stress response. Withdrawal certainly promotes relapse. And there are targets such as bias for negative affect, affective stimuli, and sleep disruption. And then one of the areas we've spent a considerable amount of time thinking about is this cognitive circuit and the disruption of decision-making. The fact that this brain disorder that evolves is heavily invested in the problem with craving, impulsive drug use, and or compulsive drug use. And there's certainly targets there that could be addressed: cognition and metacognition, interoceptive awareness of behavior, and effective connectivity. Some of that work also going on here. So we have a bit of the landscape with regard to mechanisms and neurocircuitry. And on the flip side of that is, how are we applying the information we know about mechanisms and neurocircuitry to treatment? We've had a lot of discussions today about this, and on the flip side of it, we actually do understand something about treatment, I think. And please counter me if I'm wrong. We do understand that if a continuing comprehensive care for substance use disorder recovery is applied, it can be very effective. Psychosocial, medical approaches, community support, and even medication-assisted therapy. But there are many barriers to this continuing care. For an individual, maybe they're not ready, or they feel they're not ready, or they feel they don't need it. There's a great deal of stigma associated with this disorder. It's very hard to get past that. Fear, and then in some communities, maybe not here, but many communities have very limited effective treatments. The availability is low. There's no insurance, there's no money. And oh, did I mention barriers? The barriers go way past the ones I've listed here. We do have some effective medications, buprenorphine and methadone, and I don't know, you'd probably argue plus or minuses about naltrexone, but MAT is available for opiate use disorder. The only problem is it's not really accepted and it's not employed very often. So the acceptability and availability is very low. On our campus, we have 2 million visits of patients a year, and there are 4 physicians on our campus that are MAT certified. And how many use it? None. Why? Because it's another medication, it's another drug being used to treat a drug, and that bias, that stigma associated with medication-assisted treatment is still around. Nearly all states lack MAT capacity, and even on MAT, there's still challenges such as withdrawal, events, and craving. So there's a great deal of challenge with this, even in the arena in which there are accepted, FDA-approved medication-assisted therapy. Then move to cocaine, to methamphetamine, and there are no medications there. So one aspect is to think about therapeutics. We need greater access to medications that are approved and effective. The challenges are very well described by Nora Volkow. Anybody that's seen her speak, these are some of her words. Recovery is very, very hard. Patients need every medical tool possible, and we're not giving them all of that because we don't have access to it. She also recognizes that doctors need the key tools to encourage diagnosis and treatment. Just like high cholesterol or high blood sugar, the doctor knows exactly what to do under those circumstances. In 2000, the CASA Center at Columbia did a survey of 12,000 primary care physicians. I'm not seeing this survey repeated, but those 12,000 patients were given a piece of paper that had a description of a patient who was an alcoholic teenager. 17% of those 12,000 physicians correctly diagnosed the problem. 17%. When they were then asked whether they believed that treatment for alcoholism worked, about 12% said that it worked. They were asked if treatment works for drug dependence, 4% said it worked. If you can't diagnose it, you can't treat it, what do you do with that? So we are not giving physicians what they need. And one of the things that Nora says is that you're killing two birds with one stone, providing tools to patients for recovery and tools to physicians to incorporate substance use disorder treatment in their practice. So coming from that perspective, and I am recognizing the fact that behavioral therapies are very, very critical, but can we provide medication support as a component of that? So in that line, we started based on some mechanisms of action studies that we've been conducting for a long period of time. We started thinking about how do you get from point A to point B? How do you go from a mechanism to a medication? And we needed a lot of experts to do this. But this is has anybody ever seen a target product profile before? So this is the starting point. If you are a company or you're an investigator that would like to begin to think about this is the first thing that's suggested, is to develop, and this is a hypothetical target product profile or a TPP. So the concept here is, what do we need for cocaine use disorder? So the product description is, we wanted a small molecule named, I just made this up, CURE-CUD. We identified the mechanism of action, and we wanted to reverse this dysfunction and restore endogenous brain function that's been impaired. There are details about clinical pharmacology. Ideally, it's orally bioavailable. You only have to dose it once. There's no active metabolites and high rates of compliance and retention because there's no or low adverse effects. And then the indication here is treatment-seeking adults that meet DSM-5 diagnosis for cocaine use disorder with no other axis 1 disorder. Here are primary efficacy endpoints and secondary efficacy endpoints. So from an FDA perspective, a minimal perspective is negative cocaine neurons for 30 days. We might want to actually outline a target of up to a year. And very optimistic is could 5 years result in negative cocaine neurons. Now, nothing's ever shown this profile to date. Secondary efficacy endpoints include restoration of psychosocial function, diet, sleep, exercise, reintegration, and resistance and absolute control over cocaine. This is an example of a hypothetical TPP. And it's really hypothetical because it hasn't been achieved for CUD, and it's a very, very high bar. It's rarely achieved in substance use disorders, and it has not been achieved in cocaine use disorder. This is my perspective of what we're missing. We need integrated analysis to definitively disrupt this field. And that includes, from the perspective of psychiatry and behavior, the molecular targets. We really are only seeing the beginning of data science analytics into this mix. Therapeutics discovery has been going on for a while, but we really don't know what our targets are. We know some cellular biology about the system. We have great preclinical models in some cases, great human science opportunities, human laboratory work. And then, of course, we're lacking a really coordinated education process and a consistent approach to healthcare across all of our organizations. So you can look at this list, and I've only added some of the components here as examples of what I really feel we need to do. I'm going to mention some of the human laboratory work we're doing in terms of trying to understand an addiction biosignature to use in clinical trials. I'll mention how some of our preclinical models have been driving us towards the concept that we might be able to develop some medications for therapeutics in addiction. And that's only a very small fraction of what we need to accomplish at this point. Let me go back to the target profile, product profile. When I put this together with this entrepreneurial course I was taking, I felt not very satisfied with what that outcome was. There really was to think about all the clinical trials that have failed with CUD, and then to think that we would have a target product profile. One thing that's very evident is, how do we actually verify efficacy of the treatment? What does that mean? A cocaine negative urine is one way to validate that. That's a very high bar, as I mentioned. So part of the problem, and we've talked about this today and yesterday, we have a profound heterogeneity in the clinical studies that we run. We usually identify them as DSM-5 diagnosis, CUD or OUD, with a certain mild, moderate, or severe. and what drug they're using. And there's a urine screen involved to figure that out, and maybe many different drugs that shows up in that screen. And this results in a very poor signal detection, so that we have, because we don't have a single etiology of a disorder, we don't really understand its progress, we don't know how to resolve it, and we can't predict subphenotypes, so that we can never predict get a relationship between the phenotype and what the medication is going to have, how the medication will affect that. And this obviously hampers the progress of new medications and therapies. So we have Jerry Moeller, Lori Kaiser Marcus, and a group of our team has a U54 and a HEAL initiative proposal, a funded proposal with NIDA. And NIDA developed our work group to move forward what they're calling SUD fingerprints. And it's based upon RDocs, the research domain criteria for mental health disorders. And the idea is to develop a standardized baseline platform for NIDA clinical trials. So what NIDA is trying to get to is here, if you want to do a clinical trial, here's the platform that we want you to use from a behavioral perspective. So that there is actually evidence that is available for the signature, the biobehavioral signature. Now the bio part we're still working on, but here's the base of this. We're calling it the PhAB. It's the NIDA Phenotyping Assessment Battery, and it's a group of assays and actually self-reported psychometric scales. They're the same domains that you'll recognize: cognition, reward, negative emotionality, interoception, metacognition, and sleep. And these are tasks that were already ongoing in our clinical studies. And we added a few additional tasks, all of which are available online, and they're not expensive. They're not expensive to access. The ultimate goal, and what NIDA had approached us with, was that we would establish this NIDA PhAB as the beginning parts of appreciating the behavioral phenotype, and then be able to apply, take the data and apply more statistical analysis ultimately towards the concept of getting personalized healthcare. We're a long way away from that relative to some of the other fields. However, I think it's a big, bold schema that I'm certainly on board for. So I'm just going to tell you where we are right now. We administer the PhAB as a set. So far we have 150, we've completed 159 healthy controls, 166 opiate use disorder, single opiate use disorder, CUD, also single, 76, and cannabis use disorder, I have about 125. We also have subjects that have OUD and CUD together, diagnosed for both, but the first set of analyses are going to be on these groups. And we have fMRI resting state on a cross-section of all of these participants. So far, I can tell you from a demographic perspective, they've between 18 and 65 years, the mean age of 40. We have equal men and women, which for a clinical study of this sort, we're pretty pleased with that, because typically our studies have many more males than females. The total PhAB assessment time is 4 hours and 45 minutes. Anybody here do human laboratory work? That's a very long time. And so we actually split that a PhAB assessment time into 2 sessions, and 87% of them completed the PhAB in 2 sessions. And then some of them had to come back for a 3rd session, and then again, some of them we have resting state fMRI on some of these individuals, but not all of them. So this covers, as I mentioned, multiple domains, and the whole idea is, can we parcel out either drug-related or just SUD-related phenotypes relative to controls that could give us an opportunity to then assess medications and behavioral therapies in categories of individuals. So this is an ongoing trial right now. We're still collecting a few more subjects in each of these groups, and the statisticians are working on the analysis right now. But the goal, and the reason that NIDA wants us to do this and why it's so important, is the failure rate. So we conduct phase 1 and phase 2 trials. The success rate in a very small number of participants can be fairly high, but it drops in phase 2, phase 3, and it really isn't until later that after the validated medication is gotten into a larger population that the success rate increases. So what we're hoping is that the success rate on these phase 1, 2, and 3 trials especially will be much greater once we have actually parceled out these individual biobehavior signatures in the subjects that are screened into the studies. So ultimately, what we would like to see is that rather than just only thinking about this primary efficacy point, which of course is a positive, is that the PhAB profile would predict the medication responders. Right now, we always have responders and non-responders, but we don't really know why one group responds over another. This is really relevant, and it's certainly, and I'm going to say this again, I certainly believe that behavioral therapies are a big component of treatment for addicted patients. I really do also believe that if we could target medications to a better extent to some of these biobehavioral phenotypes, we might be able to achieve what is one of NIDA's most important goals. This is an article that Kurt Rasmussen, David White, and Jane Ackery published last year in Neuropsychopharm, and it was identifying their medication development priorities in response to the opiate crisis. And if anybody, I don't know if there are any HEAL grants here, but there are that's what you got, yeah. And so you can,
It was super fast. Now we have to do it.
Now you have to do it. Definitely, now you have to do it. So there's a plethora of them. Many of them are directed at medications development. And NIDA actually published this table with a number of targets in here. And I think it's important that they also said there's unlikely to be a silver bullet among these mechanisms, which I think all of us would agree with that. And medications, I think an important way of thinking about this is that medications with these mechanisms of action are likely to be useful at different stages of the addiction cycle. So would you start somebody when they're going through recovery and early withdrawal with one medication, a longer period of withdrawal? Is there a way to stair-step that? We don't have any conception of those kinds of aspects of treatment yet. So many people in this room and many people in our group have been studying mechanisms of action. And in fact, so the drugs I just showed you on that list were all based upon, really strongly based upon the basic science that's been done in cells and tissues and in animals, and looking at these circuits and these targets. So all of these systems are on the list of medications that NIDA had just put. But they're very specific for manipulating certain aspects of the circuitry and the mechanistic target. So what we have done in our group is we have recruited a number of investigators from industry and from that are clinicians from industry and also basic scientists, preclinical scientists. And we've established two sets of molecules. The first set are what we're calling clinical assets. And these are compounds that, A, have been in humans and have been shown to be safe in humans, but then were dropped by industry as not being something they wanted to consider. So they're safe and tolerable. And the companies, and then we've established partnerships with Blackthorn for 24604, what they call 040, which has been shown to actually improve outcomes of alcohol, recovery from alcoholism. This is a partial mu agonist, GSK. It's a compound that Jonathan Hummel, who we hired from GSK, has published and is working on. It's also been on a number of about 300 patients. And this Pfizer compound, PhAB 5190457, this is a partnership with Pfizer. It's a ghrelin GHS1 alpha antagonist. So we basically began mining this literature and then contacting the companies. And we've contacted many more of the companies than we've actually established relationships. These are strategic alliances that we've established. We've got a couple more with molecules we're not able to share yet because the companies say we can't. But we're pursuing mechanisms to try to The core is the PhAB. We've submitted grants to NIDA and NIMH to try to identify if we can phenotype subjects in these studies, and then do we see a differential efficacy of these clinical assets. The next batch is we have a large number of preclinical assets in their early stage discovery. And these span GPCR manipulations like allosteric modulators. We're very interested in protein-protein interactions and how those might have therapeutic impact to disrupt them. We have some peptides. Neuraminidin U is a peptide that's a hypothalamic peptide that Jonathan Hommel has created a number of ligands that are patented. Delta Phos-B inhibitors, Gabby Rudenko is working on that project. BRD2, this is bromodomain 4. This is an epigenetic target. We are unique in having a group of compounds that have been developed and patented to UTMB that are inhibitors. Again, another PPI disruptor, D1 receptor biased agonists, ampapams or ampakines, and GPR52 ligands that are unique to John Allen, who we created from Pfizer. So we have a number of targets, and since there's a few serotonin people in here, I decided to actually talk about our positive allosteric modulators that we are developing as a potential therapeutic. Now, I shouldn't even say that. I'd be killed. One of my collaborators would kill me this. I don't say therapeutics. A set of molecules that we believe may demonstrate efficacy in our preclinical models to take it forward and develop that set of molecules further. So this is the mechanistic targets and circuits that I showed you a moment ago. This has been my interest for a large part of my career, which is the serotonergic system out of the dorsal raphe nuclei into the prefrontal cortex, the nucleus accumbens, the VTA, and how serotonin regulates that system. We became very interested in the 5-HT2 receptor because the system is very sophisticated. Serotonin interacts with 14 different receptor subtypes, 13 of which are actually G protein coupled receptors, all of these, and one ion channel, the 5-HT3 receptor. These receptors, all, if you remember about the pharmacology of 7 transmembrane domain receptors, they all have very homologous binding pockets for serotonin. So the 2A, the 2B, and the 2C receptors. And these, the 2A receptor is thought to, agonism at this receptor is thought to be the basis for hallucinations from drugs such as LSD. The 2B receptor is engaged in an unfortunate side effect of fen-phen or fenfluramine as an anti-obesity compound, which was valvopathy and pulmonary hypertension. And the 5-HT2C receptor has been thought to be very important in a number of potential disorders. So from the chemical perspective, our chemists are very interested in how you devise medications or medication candidates that separate these 3 receptors very selectively. So we have been working with the 5-HT2C receptor for a while. It became very interesting after this 5-HT2C knockout, constitutive knockout mouse was identified and characterized by Larry Teacott. He found that in these knockout mice, there's an increase in feeding, and then they become obese and actually have a type 2 diabetes phenotype during adulthood. They also demonstrate elevated Coke self-administration and elevated levels of impulsive action and impulsive choice, which is one of the characteristics of cocaine use disorder patients who have trouble terminating or stopping the impulse to take the drug. More recently, we have been investigating localized knockdown of the 2C receptor and have reported that if we knock down the 5-HT2C receptor in the VTA or the NAC, we see an increase in binge-like eating on high-fat food, elevated Coke self-administration in the NAC, if we knock it down the NAC, elevated impulsivity, and cue-about drug seeking in the MPFC especially, but also the NAC. So out of all of this is a great deal of work that spans multiple labs. but the end point is that a hypofunctional 2C receptor system is thought to be mechanistically involved in addictive dimensionalities across food and also drug. So our goal in this project was to correct suboptimal 2C or hypofunctional 2C receptor function. The idea is that there's a regulatory site that's distinct from the orthosteric site, so it's somewhere else on the receptor, and we want to target that site or those sites to develop a way to boost the function of the 2C receptor. There are two ways we're trying to do this. One is we're looking for those allosteric sites, but the second is we're also disrupting the protein-protein interactions that control 5-HT2C signaling inside the cell. The concept was to develop tactics to tune up the signaling by looking at these two targets. And I have two really gifted chemist collaborators that we work with. Jia Zhou, who's a medicinal chemist. We're working on small molecule allosteric 5-HT2C receptor modulators. And Scott Gilbertson, who is an organic synthetic chemist, they're actually different. They're different kinds of people. They think differently about small molecules. And his focus is peptide chemistry and looking at the dissociation of protein-protein interactions with the 2C. So I'm just going to focus a few of the next slides about how we went from this point into developing these compounds. As a reminder, those that you might not realize, the signaling, downstream signaling for these serotonergic molecules, this is the orthosteric site in the 7-transmembrane domain of the serotonin 2C receptor. This is a proposed allosteric site, also located on the 2C receptor. And then from an intracellular perspective, the C-terminus of the receptor actually makes a number of protein-protein interactions. And we've disrupted and published on the peptides that disrupt the binding between the 2C receptor and P10. One of our primary assays here to establish whether something's an allosteric modulator or protein-protein disruptor is a downstream signaling pathway. We're actually using intracellular calcium release as a measure of activation of this receptor and how allosteric modulator change that downstream effect. So this is the team of Jia, his postdoc, Xiaodong Ding, Chris Wild, graduate student who just graduated, and Eric Wold, who's close to graduating, and Noel Anastasio, who's a tenure track assistant professor, as the key individuals on this team to develop novel chemical entities to optimize 2C signaling. So we needed to start from ground zero, and we needed to figure out if we could actually identify molecules that bind to these allosteric sites, that they actually demonstrate and increase the response. We don't want them to bind the orthosteric site on any of these receptors, and that includes the 2A and the 2C. Ultimately, we would like to know the big question, which is whenever anybody is thinking about medications, can we actually modify the disease as a therapeutic? That's a big ask. And of course, can we reduce any target-based toxicities that might be associated with chemical compounds? So last year, Chris Wild finished his dissertation, and this is one of his 18 papers he's published. This was the second or third PhAB paper we published, but the previous two were not specifically our new molecules. So Chris was the first. We synthesized about 100 new chemical entities. We screened them and analyzed them in vitro, and we actually were able to identify a population that have no affinity or efficacy of in any of the 2A or the steric sites, and we were able to isolate those that are PhABs of the 2C receptor. Now, out of those 100 molecules, we're very focused. This is a very focused project. We want to have 5-HT2C PhABs. So anything that has that initial profile goes into the bin and can continue to be analyzed. Anything that's an agonist, an antagonist, a 2A, acts at 2A or 2B or any of those, we set that aside. We're now mining those as well, but this first goal is to identify positive allosteric modulators at the 2C receptor. This is the starting molecule. This is actually now a Pfizer compound. It was a Pharmacia compound that was published as an antibiotic, vancomycin. It's a variation of vancomycin. It was a fairly complicated molecule with a lot of chiral centers. And the first topic of from the medicinal chemist Giajo and his team was to break it into its component points. There's components. There's lipophilic tail. The central part is a pyridine core, and the last part is a polar moiety, sugar-like moiety. Then the medicinal chemist takes this apart and begins making strategic decisions about how to change this molecule. And it is our goal, so from the biology, from the in vitro lab, it was our goal to take these compounds and determine if we actually could identify 5-HT2C PAMs that were distinct from acting at the orthosteric site. And again, we used calcium mobilization to identify those. So this is sort of a cheat sheet as a publication from some of the leaders in allosteric modulation in Australia. If you look at a functional response, these are all functional live cell assays. You look at the response, an agonist alone gives us a sigmoidal curve. This is what's expected with serotonin. As a positive allosteric modulator, we're looking for an enhanced functional response or enhanced binding, which would be a leftward shift. These actions here would be either antagonist or negative allosteric modulators. Anything we see that does this, we bin them and we hold them for later analysis. So this is an example. These are very, very potent compounds. This is a compound known as, in Chris's paper, is compound 16. We're looking at the percent 5-HT induced calcium release on this axis. Black is serotonin alone. And serotonin is, if you go from 1 pico, 1 nano to 1 micromolar, you see a concentration response that is consistently in our hands, always an upward shift, an enhanced functional response with very little to no evidence of enhanced binding, which would be to the left, just curve shift to the left. We also screen them in stable expression cells with human 5-HT2A receptor and also 2B receptor. And these are the same concentrations of compound 16 that do nothing to 5-HT2 response to serotonin. This would be characterized in our hands as a 5-HT2C PAM, no 5-HT2A PAM activity, and no 5-HT2B PAM activity. This is this is actually our compound analysis flowchart. This process is milestone driven at every point. And the last point is after we get to a certain point, we then do in vivo pharmacology and preclinical models of behavior. We have, I don't know, more than 40 different assays. We have not, and I'll explain this in a moment, we do not run this entire panel on every single molecule, and we didn't even run this whole panel on our first, quote, lead molecule. But what I'll show you here is some of the evidence, since we knew that the 5-HT2C receptor was involved in the addictive dimensionality of food and drug. We tried a binge-like eating paradigm. We looked at self-administration of cocaine and oxycodone. We've been studying impulsivity for a long time. We analyzed it in the one-choice serial reaction time task. We also used drug discrimination as a proof of concept analysis to determine if our compounds look like positive allosteric modulators in a rodent. So let me take you back. This is the original compound, PNU 69176E. This is the compound that Chris identified as a lead in his paper, Wild 2019. This is the example I showed you of compound 16 illustrating a PAM response. So Chris was very interested in the potential anti-obesity value of these compounds. So the first thing we actually analyzed was, could we suppress the addictive dimensionality of in a binge-like eating model? And so what we have here is a very targeted way to approach this. This is actually binge eating. And so the way that works is you can give an animal high-fat food and they will eat a lot of it. But if you just give them a couple of hours of it and take it away and give them a couple more hours of it and then do that again, they eat a lot, even more than that. So it's a binge-like intake, and it bounces really pretty high. And I think that Smilla's been doing some of this as well. So binge eating is an elevation of high intake levels of food. So we use a dose of a selective 5-HT2C agonist, which is WAVE 163909 at 0.5 mg per kg. And this is at a dose that has no effect by itself. We gave 0.5 mg per kg of CYD 179, and there was no effect of this at this dose. And in fact, these doses are completely innocuous in every assay we've ever looked at. They're both very low doses. When we put them together, this is actually a highly significant suppression of binge eating. It's very hard to suppress binge eating in a rat. They really, really enjoy this sweet fat food. So this was a proof of concept that CYD 179 can potentiate the ability of 5-HT2C agonists to suppress binge intake. Well, then Eric Wold comes along and becomes a graduate student in this project. And he looked at this molecule and he said, I really think that what we need to do is we need to put a ring structure on this end. And so he made probably another 20, 25 compounds initially with this ring structure. And we have retained this component of the molecule, which was efficacious in Chris Wild's hands. And so here's this is compound 12. It's also has a real name, which is using one of Chris Wild's compounds, CTW0415. And here you can see we get the same upward shift with compound 12, and this is 1 nanomolar of the compound. So the first thing we do with these compounds, make sure they don't disrupt rat behavior of any kind. So we look at them across multiple doses, and we have not seen any overt effects of either compound 16 or 12 in any of our assays. This is a drug discrimination assay. Does anybody know this assay at all? You train rats to you train rats to recognize a full agonist, 2C agonist versus saline. And then once they know that discrimination, you ask the question, in this case, we've asked the question, if we take a low dose of the training drug, in this case a 5-HT2C agonist, and we combine that with a low dose of what we think is a potentiator for the 2C agonist, can we see a potentiation? So that's what's shown here. The dots are individual rats. And what we gave here is a dose of 0.5 of WAVE, the 5-HT agonist. And as we add an increasing level of the compound 12, we get a full 100%. This feels like a 5-HT2C agonist, and all but 1 animal said that. We can completely obliterate it by blocking the 5-HT2C agonist. So we're working on 5-HT2C mechanism. We've also, so that was the 2 things I just showed you were potentiation of exogenous agonists. We also have data that suggests that that the drugs themselves have effects alone. This drug itself has an effect by itself. Studying premature responses, which is a measure of impulsive action in the one-choice serial reaction time task. We get a dose-related effect of CYD19 suppressing impulsive action. And again, I'm going to mention, because this is important, there's no effect on motor activity of CYD179 up to 5 mg per kg. That's where we went. That's as far as we went. So we have a compound that is suppressing impulsive action at much lower doses than those that we went up to that were still inactive on motor activity. And then, of course, everybody in here knows this assay. Our variant is to train rats to self-administer drug, lever presses, and then achieves the drug and the cues. Most of our studies are conducted with forced abstinence. And the reason is that humans in our clinical studies don't ever go through extinction. So in thinking parallel, and we could have discussions and arguments about this, but from the human perspective, thinking about getting any of our compounds into humans, we opted to always look at forced abstinence. Usually it's in the first few days after we've established stable self-administration. And then Q-evoked drug seeking is measured. Typically, we do not measure it in the presence of any kind of priming dose of the drug. So in these studies, this is a CYD 179. This was the last small study we did with 1 mg per kg of CYD 179, which suppressed lever pressing for Qs after termination of self-administration. So to sum this, what we found with CYD 179 was that by itself it suppresses drug seeking, both in coke and oxy animals. It suppresses impulsive action, and it doesn't have any effect on motor activity in the ranges we've looked at. It also potentiates binge-like eating and interceptive effects of the 5-HT agonist in drug discrimination. So why have we terminated this project with CYD 179? Everything looked good. It was blocked by a 2C agonist. And now we had 179, which we published as what we hoped was our lead compound, and we were getting ready to do DMPK on. And then Chris and Eric showed up with CTW 0415. So why wouldn't we just go with our first molecule? The reason, so the next step in this process is to determine if there are any off-target interactions. And Brian Roth runs an NIMH-funded program called the PDSP, which I've just blanked on, Pharmacology Drug Specificity Panel or Screen, yeah. It has over 40 targets in it. And so these are targets here. And this is percent inhibition. And the way this works is the drugs, this is all a cellular assay. There's one concentration of the drug administered, it's 10 micromolar. And if anything has displacement above 50%, that's what you're seeing here. So CYD 179 displaced at 10 micromolar, which is a really high concentration, but at 10 micromolar displaced binding to 7 targets. These included the dopamine D3, the dopamine transporter, norepinephrine alpha 2A, 2B, and 2C, histamine H2, and sigma. Those are a lot of CNS off-target effects. And some, actually, we've presented this a few times, and there are some individuals that think we need to consider this as an antidepressant, perhaps. We have not done that. We basically halted the approach we were taking with CYD 179, and we immediately profiled CTW 045 because we began to get a profile that looked very similar. And what you'll see here, this is the panel. These are all the receptors. This is the percent inhibition seen at 10 micromolar. And the only one that really stuck out, and if Brian sees this as if there's anything greater than 50% inhibition, he will then actually establish a KI. So he actually was able to establish a KI for the norepinephrine transporter 2.5 micromolar. So that's the only off-target in his panel. So we felt that this was a cleaner, for a proof of concept, looking for a 5-HT PAM, we thought this was a better choice. And Eric has gone on, we have a paper that's been submitted. Eric has gone on and we have profiled CTW 0415. It has actually a very positive, acceptable PK profile in terms of all of the characteristics we look at. It's, as I showed you, it has improved selectivity over other targets in the PDSP. And Eric's his current paper and his next paper are new chemical modifications, and these are currently finalizing structure-activity relationships, and then we'll conduct a panel of in vitro and vivo analysis. But Eric didn't want to leave it there. One of the things that had the third paper we submitted, they asked about modeling. Where are these binding to the receptors? So Eric and our specialists on campus in structural biology and modeling, luckily the 5-HT2C receptor structure was identified in 2018 and published. This is the registry. This is where you find it online. Eric developed a model from this PDB file, and you refine that structure. You fit 5-HT into the model because the original model had ergotamine, this big ergot molecule in it. So he removed that structure through these computer programs, modeled 5-HT, fit the PhABs into the model, and then docked a number of other pharmacophores to that third model. And what we found is this is 5-HT in the orthosteric pocket here, and there are 7 structures here that were all positive allosteric modulators for serotonin. This is the external face, this is the extracellular loop 3 here, and they're all aligning very nicely with an extracellular allosteric modulator space. So this actually, we just got the reviews back yesterday on this paper, and this played very well. They had a couple of questions about the way we model it, but it's consistent with a single site and not multiple sites for allosteric modulation. What we haven't done is we have not docked some of the 2A allosteric modulators we have in 2B to see how those contrast. That's to be done. So in summary, we have identified 2 CPAMs. They're distinct from agonists. They may actually provide inherent or emergent advantages over agonists. We've done in vitro, in vivo, and in silico studies, and we have identified PhABs. Not all of the compounds we looked at were all 2C. We did find some 2B and 2A. We've managed to investigate in a number of behavioral studies, so we have a sense of what the profile looks like. And we've started this active-like structural model that will predict the consensus site. So as I mentioned, Eric's now working on these new scaffolds, the scalability, how we build it up, making sure the isomers are pure. And if this passes all the tests, then we now have a lead compound out of this series. So hopefully you can see how ultimately we would like to use the biobehavioral signature we've worked out to look at these new molecules that we've discovered and characterized to determine if they might have any efficacious outcomes on behavior in addicted populations. And I'm very engaged in the community, and I really believe that we actually do have a vision for the future in this arena. For the first time in my career, this is actually from the Facing Addiction in America report, which was truly landmark. But we are changing the conversation. We are having a conversation. And it's not just say no anymore. It is actually a conversation of filling that gap and figuring out what we need to establish to take our knowledge from all of our laboratories into the clinic and change the lives of people. I've been very lucky in engaging a group of donors across the country in developing a 501c3, which is named Life Insight Biotherapeutics, and it's a visionary. It's like your visionary Garvey, who sees that there's promise to the science we've created, and he is collected his colleagues and his business associates and started Life Insight Biotherapeutics as a nonprofit organization to develop some of the science and take it towards therapeutics. So I'm very proud to be involved in that. And then ultimately, although I used a lot of I's and me's while I've been standing here, these are the people that are intellectually my partners and intellectually engaged. There are students, postdocs, faculty in this plan, and I think I've mentioned John Allen and Noelle Anastasio. Kelly Dineley is a neurodegenerative. She's in neurology. She's very engaged in our project. Scott's our organic chemist. Jerry Moeller, who's indispensable in our work. And Gia Zhou, a large group of Center for Addiction Research, University of Houston Chemistry Department, and MCV, and also the UT Health Science Center in Houston. And we've been very, very lucky and blessed in being able to attract the funding that we need to keep this going. So I appreciate your time. I hope you enjoyed the trajectory of our basic science and also our clinical science perspective, and I'm happy to answer.
Any questions for Kathy?
I'm curious if you have any plans to integrate genetic information with like behavioral study through your machine learning models.
So we take from every human subject that our human participant learning, human participant that we pick up in any of our studies, we take blood and urine from everyone else. And we have been looking at some SNPs, and we've identified, that's actually, I didn't share that with you, but one very common fibrosis, we published on fibrosis receptor SNP that predicts high levels of pure activity. Those in the study that have had that. So we're collecting those data. We're not going to be serious, huge genetic screens on any of these patients. So, but that is part of the biochemical signature that we need to identify, right? So ultimately, that has to come together. So how do we capture all of that information along with the neural phenotypes and medication response in the same subjects? I don't think it's impossible.
Could you describe your self-administration assay in a little bit more detail with the cocaine, giving animals access? Is it extended access? And for looking at that population, you see differences between how much individual differences between the way they respond.
So in the cocaine self-administration study, these are all rats. And these are studies that we established that actually have a very stable level of intake across all animals. There's not a lot of variability. However, when we began doing fentanyl and oxycodone self-administration, we actually have built in some variability on short and long access. And we do see, for the opioids, we do see a broader range of individual differences. So we're actually characterizing intake by milligram per kilogram per animal and analyzing it that way. And we are finding distinct differences across animals. So in a group of 16 animals, we will have maybe 25% that are low intake, and the rest will be higher intake. That goes away in long axis self-administration. Now will ultimately level out, and they obviously become tolerant. And now they're all taking it about the same level. That's a 6-hour, 0.1 mg per kg perfusion of oxygen.
Some investigators are going to a pattern where they look for an animal of choice between food and drug. And they really want to be able to see a shift. Where a medication that would suppress both may not have as much therapeutic compliance as a drug that would shift the balance to food over cooking.
But, so this is a very interesting question, and I beg to differ with you.
Okay.
So the reality is that some of these targets, and I can name half a dozen of them, are controlling, they're titrating reward of all sorts. So food, drugs, ghrelin, ghrelin's another example, we're working on that one. Leptin, all of those peripheral metabolic regulators, they regulate food and they regulate drug intake. So what is the appropriate control? And is it a negative? So one of our opiate participants are often overweight. So in that case, maybe the utility of a compound that is controlling food and drug is not a negative. However, with coke, they are not overweight. So that might not be a positive clinical outcome if a compound that you're using for chronic treatment is suppressing body weight at the same time it's suppressing drug.
I'm not sure that they all use highly palatable food in their choice, although that certainly be one variant. So the question gets to what extent would a drug that actually does what you say, or suppresses both, actually produce a state of dysphoria or anhedonia for the animal, and maybe for the individual? Are you concerned that you might get a problem of compliance if the medication produces a state of unresponsiveness to any food?
Well, I mean, I think there are a number of strategies we can look at anhedonia and negative affect in animals, and we have not done that yet. It's on the list of things to do. From a clinical perspective, I think there are other people in this room that have a better sense of what our target profile might be ultimately.
When I thought about impulsivity and that you mentioned it also binds to the net, I was wondering, does this drug act as an allosteric modulator or a norepinephrine transporter? And, might it have effects on other kinds of impulsivity, which might even be a prodromal risk for developing drug addiction rather than a manifestation of addiction?
Yes, so that's really good. So we've made, as I mentioned, lots and lots of molecules, and we have zeroed in on this one. And if the PK continues to be as promising, that's always something you have to look at. And that's where we are right now, is making sure we're sustaining activity, or target activity, but also a good, we need to get in the brain, and these are getting in the brain. So, but once we know that, this might be, we're very, very excited about the prospects of this one being the one we can ask more detailed questions. We can address questions like Charlie has. So we're not at that point yet.
Oh, there's about 12 questions.
I had,
Let's go. Okay, Paul.
So your list of drugs at the beginning, you had some which had undergone safety testing. Using those in drug trials.
We have a grant.
No.
Yeah, where we've got a grant ongoing for the PF compound, that's the PHS, Relin GHS and Tyrus, that was one of those preferred targets. And we have a lot of that platform compound, and we're finishing up preclinical studies that look really promising. That hasn't gotten, it's been in humans and Eli Lilly, and now it's, if everything comes to CUG3 mechanisms, we have to prove the night that everything's in line with that. We're starting preclinical studies with GSK. The GSK compound, we have another compound that we're also working on, 2 other compounds. So there are a lot of these target mechanisms out there that are really pretty interesting and exciting, with profiles, that nobody's ever seen before. So I'm pretty happy with being able to start looking at these compounds and the models that we made for
On that list, you didn't have any drugs that are currently used for FDA approved alternative indications.
Yeah, so we do, but we don't need to profile those, and we don't really need to take them through the whole system. There are a number that are FDA approved. We're not working on either. We're working.
You don't work on that fast track.
Yeah. Yes. We were working with both faster, but this one.
Let's see if we have one more question from a trainee, perhaps. Then we can have, if not, there's a couple back there, and then we can discuss it in a smaller group after. Why don't you love 2 questions from 2 trainees?
Sorry.
I was just curious about PhAB 179. If you have any, have you dropped preclinical push altogether because of those effects that you saw in the pharmacological testing?
So there are 2 reasons that we sidelined that one, because that was philately, they recognized that was philately. That was worrisome to us, and that probably is contributing to the complex profile that we saw in the profile of other targets. And so we have set that on the side because of that. Although, if somebody wants to profile that, I think it's pretty, I've had a couple of people ask me what we're going to do with it, because they wanted to think about, so you got 2 camps. You have specificity and you have sort of a broad target base. So like an antidepressant. So yeah, we aren't persuading right now.
Yeah.
So I always forget what a liberal part of the country we're in here in Seattle. Like all of our psychiatry residents go through the X-waiver training to be able to prescribe Suboxone, and we do it regularly on our addiction disorder patients. And I'm just wondering, locally in Texas, obviously there's a culture difference. Is that hold up in the legislative side or more in the, clinician culture side?
It's the culture.
Okay, and what I wonder, what implications that
and
has
if
for
you
your
ask-
research in your local area.
Yeah, so if you ask them why they won't prescribe, they give you these really wonky answers, like, I don't wanna have to worry about making sure that nobody steals it, I don't want anybody coming into my clinic and trying to take you, I mean, crazy stuff like that. So we actually have a youth waiver conference at UTMB that we've got some money to put on next week. And it's mandatory for all medical students and all residents. And we also have, following week, we have the mandatory naloxone training, which, by the way, is available in CVS in Texas. You have to pay for it, it's not cheap, but you have a prescription. But we don't have it at sites all over, it's not like up here. So yeah, it's not a harm reduction study at all. Give them condoms, they'll have sex.
But maybe some more questions we can handle individually, but I want to thank Catherine for joining us today. Go on, but I think